All right, everybody, get ready for another deep dive. Today, we're going right to the core of biology. We're talking cells, how they reproduce, and how traits get passed on through generations.
Yeah, it's pretty fundamental, you know. We're going to explore DNA, chromosomes, all of that. Mitosis, meiosis, the works.
I mean, just think about it, like a single cell dividing and multiplying to create, well, anything.
Yeah, absolutely. A human, a plant, a little fruit fly.
So where do we even begin with something so complex? Like, how do cells know when to divide?
Well, they follow a cycle.
A cycle.
It's called the cell cycle. It's like their to-do list to make sure everything happens in the right order, you know, like growth, duplicating their DNA, and then finally splitting.
So it's not just random, like whenever they feel like it.
Oh, not at all. There are checkpoints, regulation. The cell cycle is split into two phases, actually.
Okay, so what are the two phases?
Interphase and the division phase.
All right, let's take interphase first. What exactly goes on there?
It's all about getting ready, preparing. Think of it like the cell is getting ready for a big party.
Okay, I like that analogy.
Yeah, so they need supplies, everything's got to be organized, right? During interphase, the cell grows, makes new proteins, organelles, and most importantly,
Replicates its DNA.
you got it, it makes a complete copy.
I mean, why is that so crucial, making a copy of the DNA?
Well, every new cell needs its own complete set of instructions,
Right.
like a recipe book, you know, to build and maintain the whole organism. The DNA's got that blueprint.
That makes sense. So DNA is duplicated. What next?
That's when the real action starts, the division phase. The cell splits into two daughter cells.
So each with a copy of the DNA.
An identical copy, yeah. There are two ways this can happen, though. Mitosis and meiosis.
Okay, I've heard those terms before.
Yeah.
I'm a bit fuzzy on the difference, though. Can you break that down for me?
Sure. So mitosis, think of it like making clones.
Okay, clones.
Exactly. That's how most cells in our bodies divide. So the new cells, they're exact replicas. It's how we grow, repair tissues, you know, the whole maintenance thing.
So, like, if I get a cut, mitosis is happening to replace those skin cells.
Precisely. And it's happening all the time, replacing old cells, keeping everything running smoothly.
Wow. What about meiosis, then?
Meiosis is a bit more specialized. It's all about sexual reproduction.
Okay, sexual reproduction.
This is how we get sperm and egg cells, those unique cells
Yeah.
that combine to make a new individual.
Oh, so this is where the genetic mixing happens, to create variation.
You got it. Meiosis shuffles the genetic deck, makes sure everyone's unique. 2 rounds of cell division, and get this, the number of chromosomes gets cut in half.
In half, why is that?
Well, so when the sperm and egg meet, the embryo has the right number of chromosomes, not double.
That makes sense. Okay, so I'm starting to see the big picture here. Can we zoom back into mitosis for a second? Like, how does that actually happen?
It's a step-by-step process. Think of it as a really well choreographed dance.
A dance.
Yeah, chromosomes are moving, lining up. There are 4 main phases: prophase, metaphase, anaphase, and telophase.
Those are some big words. Can we break those down a bit? Like, what's actually going on in each of those phases?
Sure. Let's start with prophase. This is when the chromosomes, they condense. They become visible under a microscope, like little X's. Each one's made up of 2 identical sister chromatids, basically copies of the original chromosome.
Prophase, they become visible. Okay. Then what?
Then we've got metaphase. All those chromosomes, they line up right in the middle of the cell.
Like in a line.
Exactly. Imagine them like students all lined up in a classroom. Spindle fibers, those are like tiny ropes attached to each chromosome.
Metaphase, organization, alignment. So what about anaphase?
Anaphase is where the action happens. The spindle fibers pull, separating those chromatids.
Wow, so they're pulled apart.
Yeah, to opposite ends of the cell, like a tiny tug of war, you know? Each side ends up with a complete set.
Amazing. And the last one, telophase.
Things start to calm down in telophase. The chromosomes, they reach the opposite ends, they start to uncoil, become less visible. Nuclear membranes reform around each set, and the cell starts to pinch in the middle.
So it's like the grand finale, everything wrapping up.
You got it. Last step, cytokinesis. That's when the cytoplasm divides, and we've got 2 daughter cells. Each one has its own nucleus, full set of chromosomes, the whole deal.
Wow, that's a lot of intricate steps for something that happens so fast. So we've covered mitosis, cells making copies. What about meiosis? You said it was all about sexual reproduction and creating that variation, right?
Right. Meiosis is like mitosis, but with a twist. It's 2 rounds of division, remember? That's how we get those sperm and egg cells, those gametes, which are essential for sexual reproduction.
Okay, so meiosis is a bit more complex. Where do we even start to explain this one?
It involves the same phases as mitosis prophase, metaphase, anaphase, telophase, but twice. So we go through them twice with a few key differences.
Okay, I'm ready for the details. What's the first big difference between mitosis and meiosis then?
One of the biggest happens right at the beginning, during Prophase of meiosis. Things get really interesting because this is when homologous chromosomes pair up. Remember those X-shaped chromosomes?
Yeah.
Well, we inherit one set from our mom and one from our dad.
Oh, so I have two versions of each chromosome.
Exactly. And in Prophase, these homologous chromosomes, they find each other and pair up, like a family reunion.
That's a great way to put it. So what happens when they pair up?
Get this. They exchange segments of DNA. We call it crossing over.
Oh, wow. They're swapping parts. Isn't that a bit risky?
Well, it can be, but it's also really important because it creates new gene combinations. This is a major source of genetic variation. It's part of why siblings can be so different, even with the same parents.
Mind-blowing. So they swap DNA, and then...
In metaphase I, those pairs line up in the middle of the cell, just like in mitosis. But here's another difference. The way they line up is totally random.
like flipping a coin.
Yeah. Heads one way, tails another. This random alignment leads to even more genetic diversity because when the chromosomes are pulled apart in anaphase I, each daughter cell gets a different mix of maternal and paternal chromosomes, like shuffling and dealing cards.
Wow, so meiosis is all about mixing and matching those genes, and this all happens before the second round of division.
Exactly. After meiosis I, we have two daughter cells, each with a unique mix. Then they go through meiosis tick, which is more like mitosis, where those sister chromatids are separated.
So we end up with 4 daughter cells.
Exactly. Each with half the number of chromosomes as the original, and each one genetically unique. Incredible, right?
Amazing. But what about the sex chromosomes? Don't they have a special role in all this?
They do. They're the ones that decide if an individual develops as male or female, the X and Y.
Females have 2 X, and males have 1 X and 1 Y.
You got it. And the genes on those chromosomes, they can determine certain traits, like classic example, color blindness.
Yeah.
It's more common in males.
Yeah. So it's because the genes for color vision are on the X chromosome.
Exactly. Males only have one X. If they inherit the recessive allele for color blindness from their mom, they'll express it. Females have 2 X's, so they're less likely to get 2 copies of that recessive allele.
Okay, so it's all about those X's and Y's. But what about the genes on the X chromosome? Do females produce twice the amount of proteins since they have 2 X's?
That's a great question. They don't, actually. There's this mechanism, it's called X inactivation, that ensures males and females produce about the same amount of protein from those X-linked genes.
X inactivation, what is that?
In each female cell, one of those X chromosomes is randomly inactivated, basically silenced. It forms what's called a bar body.
Oh, wow. So it's like putting one of those X chromosomes on mute.
That's a great way to think about it. It's nature's way of balancing things out between males and females. And it also explains why some female cats have those tortoiseshell coats.
Oh, that's right. So it's the random inactivation of one X in each cell that creates those patches of color.
Exactly. It's how cells regulate gene expression to make sure everything develops correctly.
This is all so fascinating. So we've covered mitosis, meiosis, X inactivation. What's next on our cellular journey?
Well, so far, we've focused on traits determined by a single gene. But a lot of traits are influenced by multiple genes, and that's what we'll explore next.
Okay, I'm definitely intrigued. Multiple genes, huh? This is getting even more complex.
It does get more complex, but it also helps explain the amazing diversity we see in nature. This is what we call polygenic inheritance,
Polygenic inheritance, got it.
where multiple genes work together to influence a single trait.
I can't wait to learn more about that.
So we were talking about traits influenced by multiple genes, right? It's not just simple dominant and recessive like those pea plant experiments.
Right, polygenic inheritance. It seems way more complicated.
It adds a layer, yeah, but it also explains why there's so much diversity in nature.
Okay, so can you give me an example? I'm having trouble picturing how multiple genes can work together for one trait.
Think about human height. There's a whole range,
Right.
not just tall and short.
Right.
That's because height is influenced by hundreds of genes, each one with a small effect.
Oh, I see. So it's kind of like a recipe with a bunch of ingredients, each one contributing to the final outcome.
Exactly. And it's the same for things like skin color, hair color, eye color. It's all about the interplay of multiple genes.
This makes so much sense. It's why we don't just see people falling into categories like tall or short. There's a whole spectrum in between.
Exactly. That's what makes it so interesting. It's continuous variation, not just distinct categories.
Fascinating. Okay, so we've talked about mitosis, meiosis, and now polygenic inheritance. What other cellular mysteries are we going to uncover today?
Well, let's switch gears a bit. Let's talk about those errors that can happen during cell division. We touched on it before, but what happens when things go wrong?
You mean mutations, right? Those changes in DNA that can lead to genetic disorders.
Exactly. Mutations are like typos in that genetic code. Sometimes they have no effect, sometimes they're harmful, and sometimes
Sometimes what?
they can actually be beneficial.
Like beneficial. I always thought mutations were bad.
A lot of them can be, yeah. They can cause diseases and health problems. But mutations are also the source of variation. It's the fuel for evolution.
Oh, so that's how species adapt and change over time.
Exactly. It all depends on the specific mutation and the context.
Okay, so could you give me an example of a beneficial mutation? It's hard to imagine a typo being good.
Let's take sickle cell anemia. It's a genetic disorder, right? Caused by a mutation in the gene for hemoglobin. That's the protein that carries oxygen in red blood cells.
Right, sickle cell anemia can cause all sorts of problems, pain, fatigue.
Exactly.
Yeah.
People who inherit 2 copies of that mutated gene have sickle cell disease. Their red blood cells become distorted. But here's the interesting part. People who only inherit 1 copy, they're carriers, but they actually have an advantage in certain environments.
An advantage. How is that possible?
Well, these carriers are less susceptible to malaria. You know, that serious parasitic disease.
So it's like a trade-off.
Yeah.
1 copy protects against malaria, but 2 copies cause sickle cell disease.
Exactly. It shows how a mutation can be bad in one context, but good in another. There are tons of examples of mutations that have actually contributed to the diversity of life on Earth.
Wow. This whole concept of beneficial mutations is really changing how I think about genetics. So these tiny changes in DNA can really have a huge impact on survival and evolution.
Absolutely. It highlights the power of genetics and how our genes interact with the environment. Speaking of power, let's talk about how humans are using genetics for our own purposes.
You're talking about genetic technologies, right?
Mhm.
Things like gene editing and cloning.
It's an incredible field, rapidly evolving. It has so much potential for medicine, agriculture, and more.
So where do we even begin with such a huge topic? What are the key technologies that have really changed our understanding and how we can manipulate genetics?
Well, one of the biggest is PCR, polymerase chain reaction. Think of it like a molecular photocopier. Scientists can amplify specific DNA sequences from tiny samples.
Wait, so they can make millions of copies from just a tiny bit of DNA. What's the point of that?
Oh, there are tons of applications. Take forensics. They
Mhm.
use PCR to amplify DNA from tiny bits of evidence at a crime scene, like a hair or a drop of blood.
It's like something out of a TV show. That's so cool. So they can analyze that tiny bit of DNA and identify someone.
Exactly. And PCR is also huge in medicine. It can detect viruses, bacteria, even genetic mutations that cause diseases. We can diagnose things much earlier now.
So PCR has really changed both crime solving and healthcare. What other exciting technologies are out there?
One that's been in the news a lot is CRISPR. It's a gene editing tool.
I've heard of CRISPR, but I don't really understand how it works. Can you explain it?
Think of it like a molecular scalpel. It lets scientists make very precise changes to DNA sequences.
Really precise.
They can target specific genes, disable them, correct mutations, even insert new genes.
Wow, it's like they could just snip out and replace genes. That sounds incredibly powerful.
It is incredibly powerful.
Yeah.
And it's being used for so much research, correcting genetic defects, engineering better crops, even fighting cancer.
It seems like the possibilities are endless. We're at a point where we can actually rewrite the genetic code.
It's a really transformative time. But of course, with that power comes responsibility.
Right. There must be ethical concerns about something like CRISPR.
Absolutely. We need to be very careful, think about unintended consequences. What if it's used to enhance traits instead of just treating diseases? There needs to be careful regulation.
It's a lot to consider. We've covered so much today, from polygenic inheritance and beneficial mutations to these incredible technologies like PCR and CRISPR. My brain is full.
It has been a whirlwind tour of genetics, hasn't it? But there's still more to explore. Next time, we'll delve into chromosomal abnormalities, those changes in chromosome numbers that can cause various genetic conditions.
Okay, I'm ready for the next part of the journey. Chromosomal abnormalities, here we come. Okay, so here we are for the final part of our deep dive. We've covered so much ground, from those basic building blocks of DNA to incredible technologies that can literally rewrite our genes. It's mind-blowing.
It is. But we're not done yet. We still need to talk about those changes in chromosome numbers that can sometimes happen. Those chromosomal abnormalities.
Right, right. Like we talked about errors during meiosis when those chromosomes don't separate correctly.
Exactly, that non-disjunction. It can lead to gametes, the sperm and egg cells having too many or too few chromosomes.
And if those gametes combine during fertilization?
Well, the embryo might inherit an extra chromosome or be missing one. That's what we call a chromosomal abnormality.
So it's not just about the genes themselves, but about having the right number of chromosomes, too.
Exactly. Imagine a recipe, right? Too much or too little of something can really mess things up. One of the most well-known examples of this is Down syndrome, trisomy 21.
Yeah, Down syndrome. That's when someone has an extra copy of chromosome 21. I know it can lead to some developmental and health challenges.
That's right. They often have certain facial features, and they might experience developmental delays, intellectual disabilities, and an increased risk for some health problems like heart defects.
It's definitely a challenging condition. But I also know there are a lot of people with Down syndrome who are living full and meaningful lives.
Absolutely. And there's more and more awareness and support now, along with better medical care that can really help.
That's good to hear. So Down syndrome is one example, but there are other conditions that can happen because of chromosome number problems, right?
Oh, yeah, definitely. There are other trisomies, though they're less common than Down syndrome. Trisomy 18, also known as Edwards syndrome, and trisomy 13, Patau syndrome. These usually lead to more severe developmental problems.
It's hard to think about those families going through that. It shows just how delicate that balance of chromosomes is.
It really does. And then there are cases where a chromosome is missing. We call those monosomies. The most well-known is Turner syndrome. Females are born with just one X chromosome instead of two.
Wow, a whole chromosome missing. That must be really serious.
It is. Girls with Turner syndrome are often shorter, might not go through puberty naturally, and can have other health problems like heart or kidney issues.
It's amazing how much impact even one missing chromosome can have.
Right. It really shows how important that balance is. And, while these chromosomal abnormalities can be really challenging, research is always moving forward. There's better understanding, treatments, and support for the individuals and families dealing with these conditions.
That's really good to hear. This whole journey exploring cells and genetics has been incredible. We've covered so much.
It's been a pleasure sharing this deep dive with you. I hope our listeners are walking away with a new appreciation for just how amazing those processes are that make life happen.
I know I am. And I have to say, I'm so grateful you are here to guide us through this fascinating world of biology. It's really a world of wonder.
Thanks for having me. And to everyone listening, keep learning, keep exploring, and keep being amazed by the world of genetics that makes each of us unique.